Pancreatic ductal adenocarcinoma genetics. Unlike the breast and other cancer, pancreatic ductal adenocarcinoma is not an easily divided into subgroups by molecular characteristic at the present time to guide therapy or to inform prognosis. Moffitt and colleagues, they performed virtual microdissection of the pancreatic ductal adenocarcinoma tissue combined with gene expression microarray data and identified two subtypes that were associated with the survival outcome. The classical subtype was associated with an improved survival, while the basal-like subtypes, they were associated with a worse survival. So classical and basal-like. There are two subtypes. So classical subtypes associated with an improved survival, basal-like subtype associated with a worse survival. Findings have not been adopted into the clinical practice or incorporated into the precision oncological care to this point. Whole exome sequencing of numerous pancreatic ductal adenocarcinoma have been performed and provide some insight. High throughput sequencing data emphasize the molecular complexity of pancreatic ductal adenocarcinoma and underscore that no two pancreatic ductal adenocarcinoma tumors exist alike. Out of 24 genome, 1327 different genes, 7% of the all coding genes were found to have at least one somatic mutation, and 148 different genes had at least two mutation, and these were on an average 60 genetic alteration per tumor, and each pancreatic ductal adenocarcinoma had a unique genetic fingerprint. Unfortunately, sequencing studies did not identify any novel high-frequency mutated cancer genes as promising therapeutic or actionable target, and a more practical approach to personalized therapy has been proposed, which group genetically altered genes into 12 core signaling pathway, apoptosis, DNA damage, 10 others. These pathways in theory may be more realistic therapeutic targets than single genes if effective targeted therapeutics becomes available. And a genetically altered gene may be categorized as an oncogene with gain of function mutation, tumor suppressor gene as loss of function, or genome maintenance gene, that is loss of function. And for the latter two genes types, both copies of the genes are typically inactivated, and one allele is lost by somatic mutation and the other by chromosomal allelic loss of heterozygosity. And oncogenic KRAS is altered in more than 90% of the cases of pancreatic ductal adenocarcinoma and is usually an early event in tumorigenesis, and this genetic change represents the only high frequency oncogenic mutation in pancreatic ductal adenocarcinoma and therefore is considered by many to be the most compelling therapeutic target. And somatic KRAS mutation mostly occur in the codon 12 and more commonly of glycine to valine. So KRAS G12V, glycine to valine, and glycine to aspartate. So KRAS G12D. And the sequencing alteration inactivate GTPase function, leaving GTP continuously engaged and the oncogenic constituently activated. So activated KRAS positively regulates multiple signaling pathway including BRAF, MAPK pathway, proliferation, PI3K, mTOR, cell growth and survival, and PLC, PKC, calcium and second messenger signaling. And recently an inhibitor of the KRAS codon 12 glycine to cysteine mutation, sotorasib, that has been developed and approved for the treatment of non-small cell lung cancer. And KRAS G12C is present in 13% of the non-small cell lung cancer but only 1% in pancreatic ductal adenocarcinoma. And given the early development and prevalence of the activating KRAS mutation, inhibitors of KRAS represents opportunity for potentially effective targeted therapy with large therapeutic window but have eluded researchers thus far. And the remaining commonly mutated pancreatic cancer gene are all tumor suppressor genes. So only KRAS is the oncogene, others are tumor suppressor gene, and the pattern of frequent allelic loss mutation in pancreatic ductal adenocarcinoma due to chromosomal instability losses are more common than gains. and promote a fertile environment of genetic experimentation that favors loss of function mutation. Allotype mapping reveals that genetic loss ranges from 17 to 80% of the genome in a given pancreatic ductal adenocarcinoma. And allelic loss is non-random across pancreatic ductal adenocarcinoma genome, and loss of heterozygosity, that is hotspots, areas where genetic loss most commonly occur, harbor the most important tumor suppressor gene in pancreatic ductal adenocarcinoma, CDKN2A, P53, SMAD4. CDKN2A is inactivated in roughly 95% of the pancreatic ductal adenocarcinoma, either through homozygous deletion of both alleles, somatic mutation combined with loss of heterozygosity, deletion of one allele, or promoter hypermethylation. Inactivation of this gene arbitrates the RB1 mediated G1 as checkpoint in the cell cycle, allow unchecked inhibitor of RB1 by CDK4, promoting cell cycle progression, cells and cancer cell proliferation. P53 combined with the loss of heterozygosity occurs in majority of the pancreatic ductal carcinoma. P53 is critical component of the DNA damage response, and in fact of cytotoxic stress, P53 induce cell cycle arrest at G1 or G2 at the cell cycle, allowing cells to repair their DNA prior to DNA synthesis or mitosis. Thus, P53 loss contributes to chromosome instability and aneuploidy observed in pancreatic ductal adenocarcinoma. P53 has been targeted experimentally by reactivating the mutant isoform or by targeting the G2M checkpoint with WEE1 inhibitor as a synthetic lethal approach. P53 deficient tumors are particularly dependent on the G2M checkpoints. SMAD4 DPC4 RNA is inactivated in roughly half of the pancreatic ductal adenocarcinoma through either homozygous deletion or mutation combined with loss of heterozygosity. And SMAD4 is a part of transforming growth factor beta receptor pathway, and like the other two aforementioned tumor suppressor gene regulate cell cycle at G1 as checkpoints. Interestingly, preservation of the SMAD4 protein expression is associated with a local predominant progression pattern in pancreatic ductal adenocarcinoma, which could be used to guide patient selection for radiotherapy. And a group of genomic maintenance genes like BRCA2, PALB2, FANCC, FNCC and CG involved in the Fanconi anemia DNA repair pathway mutated in less than 10% of the pancreatic ductal adenocarcinoma are particularly intriguing, and since tumor deficient in this pathway are highly susceptible to the DNA damaging agents and PARP inhibitor therapy. So analysis of laser capture microdissection pancreatic tissue have elucidated the chronological sequences of the major genetic changes in pancreatic ductal adenocarcinoma tumorigenesis. Telomeres shortening, KRAS mutations, they are believed to be the earliest event. P16 loss occurs at the pan in 2 stage. P53 SMAD4 BRCA2 inactivation occur later during pan in 3 stage, and major chromosome instability characterized by large scale allele copy number change is rarely observed in pan in lesions and principally limited to loss of heterozygosity hotspots. So Ayako, Buzio, Donahue and colleague extended the molecular progression model of pancreatic ductal adenocarcinoma, the most advanced stage of the disease, through a rapid autopsy program in which primary tumor and paired metastases were sequenced and compared, and the investigators distinguished founder mutation, those that arise early in the tumorigenesis and the present throughout the tumor, about 2/3 of the mutation, from progressor mutation. The founder mutation, progressor mutation mutated in the subclonal population of cells and absent in the parental clones, about 1/3 of the these mutations. Interestingly, the progressor mutations that existed throughout a metastatic deposit were also identified at a certain microdissected foci in the primary tumor, but not throughout the primary. And this finding demonstrated that the parent clones giving rise to a specific metastasis can be defined and mapped in a primary tumor, while previous sequencing studies highlighted inter-tumor heterogeneity, and this study was the first to identify intratumoral genetic heterogeneity in pancreatic ductal adenocarcinoma with the profound implications of therapy. Thank you.